Discovering Green Turtles, Chelonia mitis, Foraging Habitat in the Galapagos Islands Using Stable Isotope Analysis by Sabrina Bocek, with project partner Garrett Lemons, NOAA's fisheries biologist and scientist of the Marine Turtle Ecology and Assessment Program, manager of the Stable Isotope Lab at Southwest Fisheries Science Center. Green turtles are circumglobal species primarily found in subtropical and temperate regions of the Atlantic, Pacific, and Indian Oceans. They live to be about 70 years old and go into reproductive maturity around 25 years of age. The Galapagos Islands is where our study samples came from, and the Galapagos Islands is 1,000 kilometers away from the South American continent and is the largest marine reserve in the world. Here we are sampling putative prey items such as seagrass, algae, and invertebrates. We are also taking biopsies of the dorsal epithelial tissue in about 60 turtles in the 3 ecoregions of the Galapagos. These samples are then processed at the NOAA's Stable Isotope Lab at the Southwest Fisheries Science Center. Here's a machine that separates lipids. These prepared samples are then sent to another lab to be processed through a mass spectrometry machine. The injected samples heat up and ionized particles are sent through a magnetic field which separates based on mass. The stable isotopes that help discover foraging and trophic ecology are carbon and nitrogen. Isotopes mean that there's different numbers of neutrons in the nucleus, giving it different masses of the same element. So we have carbon, and then we have stable isotope of nitrogen that we're also looking at. Now what the stable isotopes do, the nitrogen has ranges for each trophic level. So then we can know where in the trophic ecology food web the sea turtle is getting its energy from. While the carbon isotopes tell us how near the seashore or out into the pelagic waters their diet items are coming from. An isospace plot looks like this. Here we have the diet items, and then here we have where the turtles are, and we can place the diet items and everything in trophic level, and the higher the carbon, the closer to the seashore. So we are getting all the data for the source and the mixture. We put that to the trophic discrimination factor, and we will analyze that in our software with the mixed CIDR package using Bayesian statistics.